So proteins with multiple passes, right, begin looking like this. So rather than having a signal peptide on the amino terminal end, they have what we call a start transfer sequence. And a start transfer sequence is a sequence that starts the transfer of a protein through the membrane that's not terminal. So you can see that what happens is this protein was synthesized, and a portion of it, the amino terminal end, stays in the cytosol. Then there's the start transfer, and the start transfer gets it into the membrane like that. And then eventually this next guy there, right, is a stop transfer sequence. And he stops the transfer. So now we have both of those, the start transfer, the stop transfer. This guy, since he's not on the amino terminal end, is not cleaved out. He stays as part of that. So when the translocon sort of opens up or separates or allows these to come out, whatever it does, we end up with two of these, the start transfer sequence and the stop transfer sequence embedded in the membrane. So if this were the protein, if it were to end here, we have a protein with an amino carboxy in the cytosol, and we have a loop, what we call a loop on the inside of the endoplasmic reticulum, and the lumen of the ER. We have a loop between the two spanning regions. But generally, proteins don't have two regions like this. If you look at proteins that are integral in the membrane, which means that they have portions in the membrane that transverse the membrane, there are numbers that are commonly used. One of the biggest is 7. There's a lot of proteins that have 7 alpha helices going through the membrane to sort of anchor them in there and help them with their function. The G-coupled protein receptors are like that. Another fancy number is 12. A lot of them have 12 of these helices in there. A lot of the proteins that are membrane proteins that sort of make pores or channels through the membrane have 12 of the alpha helices, and those helices kind of arrange in a round or in a layer, to make a pore through the membrane. Anyway, so they don't have just 2, they have more than that. Although this says this could be a mature double pass membrane. I don't know any double pass membranes. I guess that's all I want to tell you about. So Al, maybe I'll just tell you here. So what would happen for another pass, right? What you need is now, as the protein is being synthesized, you'd have a second start transfer sequence up in here. So even while this was in the membrane, that start transfer would get into another translocon. And synthesis would continue, and after that start transfer, later in the sequence, there would be another stop transfer sequence. And that stop transfer would end up, and it would look kind of like this. And it would open up, they'd come out, and then you'd have a protein went like this, back through again, and back up again. And you'd have 4 of them. Yes?
So these signal sequences don't get cut off?
Say that one more time.
These signal sequences don't get cut off?
No, they do not get cut off. None of them are. They function as anchors. In fact, a lot of times, one of these is called the start transfer anchor sequence. So they anchor the protein. The only sequence that can get cut off of a protein, right, is one that's on the end of the protein. We don't have a protein that really is a protein ligase that ligates 2 proteins. We do, right, but it's involved. It's the ubiquitin ligases. But as far as processing proteins, we don't have one where a protein is cut and a part is removed and then we ligate it back together, like an intervening sequence or like any of those types of things. So we don't have an RNA or DNA type ligase. We just have the ubiquitin ligase. And I'll tell you about that later, I hope. Okay, so if you look at a protein, right, you can look at the primary sequence of a protein, which we can develop, right? So you sequence the DNA, and from a DNA sequence of a gene, you can predict the protein structure. And when you predict the protein structure, the amino acid structure, you can analyze that structure, and you can run a program that will tell you whether or not the protein, where it'll look at hydrophobicity. Look for regions of the proteins that are very hydrophobic. And in this chart here, it shows you that this is a hydrophobicity plot. And it shows you groups of hydrophobic amino acids, and then when they get amino acids below the line are hydrophilic amino acids. And what the program looks for is groups of amino acids, right, where there are at least 20 of them, because it takes 20 to span the membrane. And you can see that these red regions here and these yellow regions here are regions that span the membrane. So essentially, here's the sequence here. You can see this is a protein that has a start, stop, start, stop, start, stop sequence. run it through this program, right, it will give you a graph like this, and you can determine where the membrane spanning regions are. The first one, the second one, the third one, the fourth one. The first one, the second one, the third one, the fourth one. The third one is the stop sequence. The fifth one is the stop. The sixth one is the stop. And you end up with 1, 2, 3, 4, 5, 6, 7 spanning regions. So this is like the G-coupled protein receptors and many other proteins in the cell. So there are a lot of proteins that look like this. They have an amino terminal sequence in the lumen of the ER, carboxy on the cytosol, and then each of these spanning regions are connected by loops, either cytosolic loops or well, they usually don't call them endoplasmic loops, but they're usually called extracellular loops, because that's where they'll end up. Most of these membrane proteins end at the surface. So all this says is we can look at the sequence of amino acids and predict where that thing's going to have spanning regions by looking for sequences that are at least 20 amino acids long, that are long enough. Do you see why they have to be 20 amino acids? Right? You have to have 20 in order to span the length. This is the lipid bilayer here. You need 20 of them in order to span that. If there's any less than 20 hydrophobic, then you have charged residues extending in on either side. That's impossible. They can't exist in the lipids. Okay. So what happens to proteins after they move into the endoplasmic reticulum? After they're synthesized in the ER, they're moved by vesicular translocation or vesicular movement from the endoplasmic reticulum to the Golgi apparatus. But before that happens, right, as proteins are moving into the ER, they're modified. So we said that proteins, first of all, the endoplasmic reticulum is a sorting organelle, but it has another function, a very important function, and that is that proteins, all the external proteins are folded. So the endoplasmic reticulum is a folding mechanism, a folding center, right, where proteins are folded. And one of the very first things that happens that's involved in this folding proteins are glycoproteins, the synthesis of glycoproteins. So as proteins are being synthesized and moved into the endoplasmic reticulum, if they are to be glycosylated, they are. That's where glycosylation begins. And it begins, there's two types, but at least in M-linked glycosylation, which means that this glycose, or this sugar chain, is attached to an asparagine residue, right, to the nitrogen of the side arm, the nitrogen at the R site of asparagine, right? That's why it's called N-linked, it's to that nitrogen. But there it starts when this core carbohydrate is attached to that nitrogen. So there is a specific sequence that leads to that. Probably happen in the next slide, I'll show it to you. But if that sequence is satisfied, right, it happens to be an asparagine, any amino acid, that X, or a serine, and then a serine and a threonine. So if you have asparagine, X, serine, or threonine in a sequence, that's a glycosylation site, N-glycosylation site, and an enzyme which is known as, I'll show it to you in the next slide so you can see the name of it, transfers this core carbohydrate that has 14 sugars onto that thing. So all glycosylation begins with this core that has these 14 sugars. 2 N-acetylglucosamines, 9 mannose residues, and then 3 glucoses on the end. Okay? Everybody follow that? So here's another picture that shows the same thing. These guys are coming through. There's the asparagine. These core carbohydrates are carried on a molecule called dolicol phosphate. And dolicol phosphate is sort of interesting. This dolicol is this big, large lipid, big fatty acid with like 100 residues, 100 carbons in it. It loops through the membrane, so it's anchored very tightly. And these carbohydrates are synthesized, they're added to that by glycosyltransferases. They're built on there one at a time. But in the endoplasmic reticulum, as this is coming through, at this asparagine residue, the enzyme oligosacchαρyltransferase, it's written there, I didn't have it on the other page, but oligosacchαρyltransferase transfers that core carbohydrate to the asparagine residue. There can be more than one. Many proteins have a lot of these on there. The major protein in your red blood cell is called glycophorin, and that protein is by weight is 65% sugar. So So the sugar residues weigh more than the amino acid carbons in that particular molecule. So these can really coat. And there's several functions for these sugars on the surface of cells. Oligosaccharides protect proteins from degradation. That's the biggest thing. They protect them from enzymatic degradation by proteases. So most of the proteins, probably 95, maybe even 99% of the proteins that go through the ER are glycosylated. And they end up on the surface of the cell glycosylated, which protects them somewhat from exterior or even from alien proteases, right, from different organisms that attack the cells. They have other functions. One is that they're recognition molecules, right? So there are a specific type of proteins that are called lectins, and lectins recognize and bind to these carbohydrate moieties that are in a specific sequence. Okay, so this first glycosylation, the N-glycosylation, begins in the endoplasmic reticulum. I'm going to tell you later, I guess maybe tomorrow or whenever we get there, we'll talk about what happens in the Golgi apparatus and thing. But I want to tell you a little bit about mitochondria and chloroplasts. Not a lot, but a little bit. So if you look at these things, these organelles, they present a very special problem, and that is that they have two membranes, and they have two spaces. They have an intermembranal space on this guy, and then they have this matrix, right, the inner space. These guys actually have three. They have that same double membrane around the surface, but then the stroma of the chloroplast is also another membrane system. So they not only have the intermembranal space, and they have the stroma, this region here, and then they have the thylakoid space inside the thylakoids. I think I call that the stroma. This is stroma. These are called thylakoids. The thylakoids are the stacks of membranes inside chloroplast, where all of the like harvesting enzymes and all of the dark reactions occur on that thylakoid there. Chlorophyll's in that thing. But anyway, proteins have to be targeted into the intermembrane space, into the stroma of the matrix, and into the thylakoid. So how are these proteins get in there? How does a protein get through the two membranes, right? It has a signal peptide, or does it? Turns out they do. And the signal peptides are slightly different. They're usually longer. They can be as long as 60 amino acids long. One thing that is true of all of these is that the